topic today is to application of methylation gene detection in precision diagnosis of endometrial carcinoma. Before we start, I would like to introduce our team briefly. Beijing OriginPoly, as we know, we call Poly. It's a startup biotech company, and we are focusing on the women healthy care. Our headquarters is located in Beijing and has a branch in Hong Kong. Since we establishment in 2020, some of our IVD solutions have already obtained European CE and the British UKCA certification already. Currently, we are planning to on several scientific research cooperation. Let me take for example, in September this year, we began to prepare to cooperation with the ISGYP to explore research on methylation in European. So regarding scientific research, we have established several research projects in cooperation with many medical centers in China already. And the related research has appeared in like ESCR, ISCO, ISG. We have completed the development of the methylation detection solution for gynecological tumor, including the cervical cancer, endometrial cancer, and ovarian cancer already. We have also started clinical research on the detection of reproductive trait microecology. We look forward to seeing the result and hope to learn from this study and to help to improve our clinical needs in the future. So in today's sharing, I will discuss the current research on our early screening and the diagnosis of endometrial cancer through methylation detection. As you may know, due to the factor like the increase in obesity and diabetes, the prevalence of EC is still on the rise. According to the study in China, there are more than like 17,000 new EC patients every year, and it has become one of the top 10 female cancers already. As we know, early screening and the diagnosis are essential for the EC treatment. Therefore, a better earlier screening technology is an important topic in clinical practice. However, we recognize that many challenges for the EC in the current practice of early diagnosis. Let me take for example, the present technology is still inconvenient for the large-scale EC screening because of lack of convenient non-invasive methods with high sensitivity and with specificities. Also, in the past, due to the technical and the postology limitation, the research progress of EC-related biomarker was slow. However, in recent years, we have seen that clinical researchers around the world has increased their attention to EC diagnostic technique. At the same time, in China, the system research and the development to establish the EC early screening are also included in one of the national 5-year plans already. So our research team, we are focusing on the development of EC biomarker detected through the methylations. We know that the change in DNA methylation level and the pattern has become one of the critical markers of cancer. Because methylation can maintain a stable state in the long term and can be more easily to obtain in various type of sample, especially for the clinical sample. So it can improve the availability of clinical testing applications. So as we know, methylation is a kind of epigenetics, and the change in methylation level and the pattern will have the opportunity to further turn it on or off of the downstream genes and further change the proliferation and the activation of human cells. Therefore, under the same genotype, through the regulation of methylation, there will be able to form a different phenotypes. So in the process of cancer formation, methylation is considered to take a part in an important regulatory mechanism. In some tumor suppressor gene, once they are be methylated, there's a chance to inactivate the gene. So this causes the expression of the tumor suppressor gene to be turned off and the cancer signal pathway to be further activated. Therefore, in this context, the current change in gene methylation are considered to be one of the risk indicators that can be applied to detect cancer of short-term cancer progressions. So currently, we know that there are already some IVD product or IVD solution application to cancer, like colorectal cancer, like cervical cancer already. So therefore, under such a context, we would like to explore how to use the gene methylation as an early detection technology for women's cancer of the reproduction system, especially for the EC. So we also want to establish screening methods with a high sensitivity and high specificity and with successful sample collections. So the first question is how to find the biomarker suitable for the clinical diagnosis of EC. Like the discovery of most biomarkers, we have also gone through the 3 phases, including the finding candidate gene, validation of candidate gene through a pathological specimen, and finally, we forming a complete gene IVD detection kit and carry out to clinical verifications. So among our candidates, we found 2 important genes, namely the gene methylation of CD01 and the CLF4. These 2 genes play an important role in cell growth and the development, and also it is expressed in the tumor during the tumor growth. Then we face another question: is the double gene methylation have the tissue specificity, or can methylation be detected through the non-invasive methods? That is through the cervical exfoliative active cells. So we further compare the methylation of the different tissue sites and the cervical exfoliative active cells to answer this question. And we found a high tissue specificity in the methylation of those 2 genes. At the same time, positive results was also expressed in the exfoliative active cells from the different part of a tumor patient, which means that we will have the opportunity to detect EC through the non-invasive detection of methylation and even apply it to early screening and early diagnosis. So to further verify this possibility, we conducted several clinical studies with the Peking Union Medical College Hospital team, including a single center retrospective and a prospective study, and also a multi-center prospective studies. In single center studies, 2 trials has been included nearly 1,005 clinical sample and demonstrated that the cis-endo, which means the double gene methylation, had 88% sensitivity and 93% specificity performance in EC. However, we want to verify, to do more, to verify the applicability in practice. So we further carry out endometrial, which is a multi-center clinical research. We recruit more than 10,000 cervical exfoliative active cells in this study, and based on the outpatient clinics. Our current data show that the sensitivity and the specificity of the cis-endo, which means the double gene methylation in EC, for EC, are more than 90%. Endometrial, this study is led by Peking Union Medical College Hospital and the cooperation with the 14 research center in China. It recruited 10,000 participants, and the current data suggests that the performance of using the dual gene methylation detection of EC are significantly better than the detection based on the symptoms like the image. Interestingly, we also find something different, like the methylation sensitivity at different sampling collection point will change. So this may suggest that the methylation aspiration in EC is useful for the early diagnosis and has the clinical significance in the treatment process. We are also very looking forward to do more research and the data in the future to support this and to demonstrate that this possibility. So from the data we collect for now, the data we collected, the CD01 and the CLI4, the double gene methylation detection will likely to be better than the current methods. Suggest that this methylation may be used as an independent screening for clinical EC diagnosis in the future. At the same time, in the clinical path of EC, early diagnosis and early screening, it may have the opportunity to become one of the short application to reduce invasive hysteroscopy. So we believe that gene methylation detection will have the three main clinical application scenario based on our understanding, based on our current unmet clinical needs, based on our finding during the clinical research. The first one is the screening tool for, especially for EC in patients with a post-menopausal bleeding to avoid them like a misdiagnosis and the delayed examination due to the fear of hysteroscopy. We know there's a possibility of misdiagnosis only based on the like a TVS inspection indicator, and it will be a highly dependent on the clinical like experience and the operator skills. So this will be an excellent challenge for the area that need to like screening, especially in rural area or place without a medical resource. So through a non-invasive collection methods, there will be a new opportunity to serve as a supplementary means. And recently, we have also analyzed the clinical application result of combined gene methylation and the TVS in postmenopause women. In the current single center study, we found that using the TVS alone can only achieve more than like a 75 sensitivity. However, if we can further combine with the gene methylation, which means combine with the gene methylation and the TVS, which there will be an opportunity to increase the detection sensitivity to over like 99%. Therefore, combining clinical imaging and the application of methylation biomarker will be a better way, probably be a better way to integrate medical economics and availability. Of course, we still need more data and more research to prove that. So our team currently conducting a related research already, and we look forward to more researchers to join us together. So the second scenario of methylation detection is assisting endoscopy combined with like a pathological sampling to avoid hidden or misdiagnosis of early endometrial cancers. And thirdly, methylation technology also can be used in follow-up in the management of EIN women or young women with fertility needs. For example, we observed in some small sample data that the effect of progesterone treatment is better for patients with the EIN, but the methylation has been like a negative. So we have to conduct some clinical research on this part and hope to share the results with you soon. And finally, we also propose the application of methylation as a management and follow up the EC through the whole disease progressions. That's to say, the qualitative results of methylation are further extend to numerical like quantification and can be used as one of the follow-up indicator for clinical. Such a dynamic index will have the opportunity to improve patients' compliance with the diagnosis and the treatment process and benefit patients. So such an application hypothesis, again, still need more large-scale research, and we expect more clinical research can cooperate with us. So, okay, that's it. Thank you for your time today, and it's truly a great honor to have the opportunity to share our research result with you. In the future, we will publish more clinical study results, and we hope to be seen by more people through this opportunity, and to conduct more research and contribute to female healthcare. Thank you.